Thank you for joining us today. I'm Tracy Smith, Program Director of Education and Outreach for the Center for Integrated Cellular Analysis based in New York City. In this seminar series, we highlight new papers and methods from our center that we believe to be of interest to the broader community. Today, we will discuss CPA-Perturb-seq, an extension of the Perturb-seq framework designed to study cleavage and polyadenylation, a post-transcriptional regulatory process. As always, we'll have time at the end of the talk for questions, and we encourage you to either raise your hand and I'll ask you to unmute yourself, or you can type your question into the chat and I'll read it aloud to our speakers. So our guests today are Hans-Hermann or Harm Wessels, a senior scientist at the New York Genome Center, and Madeline Kowalski, an MD PhD student at New York University. Harm did his postdoc in the labs of Rahul Satija and Neville Sanjana at the New York Genome Center, where he focused on the development of novel technologies to enable massively parallel screening of gene functions using content-rich multimodal single-cell readouts. He has recently joined the Technology Innovation Lab led by Sanya Vikovic. Madeline Kowalski is an MD PhD student in the Satija Lab. Prior to joining NYU, she completed her master's in biostatistics at UNC Chapel Hill in Yun Lee's group, and completed her undergraduate degrees in mathematics and statistics at the University of Chicago. With that, welcome, Harm and Maddie. I will hand it over to you.
Thank you for the nice introduction, Tracy. As Tracy said, today, Maddie and I, we're going to talk to you about our most recent preprint that we just put on bioRxiv, where we wanted to use Perturb-seq in order to study post-transcriptional gene regulation specifically. And specifically, I mean, we wanted to look at alternative polyadenylation. So alternative cleavage and polyadenylation is a process that occurs co-transcriptionally and that fundamentally leads to transcripts that are different at their 3' end. So specifically, there's a cleavage and polyadenylation complex that may choose between different polyadenylation sites within a transcript. So you can, for example, have alternative polyA sites in the terminal exon. So you can distinguish between proximal and distal sites. But you can also have polyA sites in alternative last exons or even intronically, which can then also lead to alternative last exons. But all of these choices lead to differences in the repertoire of cis regulatory elements in the 3' UTR, and these can, in consequence, attract a different set of transacting factors. So therefore, alternative 3' UTR usage basically can influence RNA stability and decay, for example, RNA localization, or also protein function directly. So therefore, like alternative polyadenylation has been found to play a role in a couple of in a lot of different mechanisms within the cell. So as I already said, it is part of to influence RNA stability, among others. And for example, it has been found that there's a different repertoire of the length of 3' UTRs depending on the cell type. So for example, more differentiated cells tend to have longer 3' UTRs, while more highly proliferating cells tend to have shorter 3' UTRs. And this has, for example, been identified in a cancer context where, for example, shorter 3' UTRs can evade negative regulators such as microRNAs and therefore lead to higher output of the gene products. An alternative mechanism that has been implicated with alternative 3' UTR choice is, for example, the RNA localization. And one of the examples here is, for example, that depending on the 3' UTR information that is present, transcripts may stay in the soma of a neuronal cell, or maybe be exported to the neurites, and therefore can lead to polarization within the cell, and also to different translational outcomes depending on where you are within a large cell. And then, as I said, it can also influence protein function directly, and maybe the most prominent example here has been identified for CD47. So here, depending on which 3' UTR is present, the 3' UTR can act as a scaffold and lead the transcript to be translated at a certain place and then have access to be translocated to the cell membrane. So here, for example, the long 3' UTR isoform will lead to a protein product that is going to be presented at the outside of the cell, while the short isoform, the protein product, will stay within the cytosol. So you can imagine that since alternative polyadylation is involved in many processes, that it is also influenced in a couple of human diseases. So, for example, it has been found in one of the prominent examples that structural variants in PDL1, which is a checkpoint gene and also known as CD274, for example, these structural variants can lead to increased expression of the short isoform. And since this is a checkpoint gene, this can essentially lead to immune escape of cancer cells. While this is like a larger structural variant, there are also single nucleotide variants in cis-regulatory elements that can directly be associated with disease. And here I just listed a couple of those, including a couple of thalassemias, but also cancers for TP53 alternative polyadylation, for example. So cleavage and polyadylation is, at the end of the day, regulated at the sequence level in a multi-protein complex, or the cleavage and polyadylation complex. And this complex, as I said, travels together with RNA pol2 and is itself, it is composed of multiple subcomplexes, like, for example, the cleavage factor 1 complex, the CPSF complex, or the CSTF complex. And these scan the nascent transcript for different regulatory elements in different parts of the nascent transcript. So we can basically look at upstream regulatory regions, the canonical cleavage motif itself, which is a hexamer of surrounding the sequence context of AAU, AAA, but also distal regions, which all of them can influence the choice of alternative polyadenylation. But overall, if we look at the sequence context across all polyA sites, we see a characteristic signature of sequence composition around the end of transcripts. But while this is like some sort of composite across all sites, we can also further zoom in and look and try to distinguish what is different between different sites. And then we can, for example, see that in proximal polyA sites versus distal polyA sites of tandem 3' UTRs, we, for example, have a different prevalence of the canonical cleavage motif, or motifs such as UGUA, which is usually recognized in the cleavage factor 1 complex, that they have different prevalence depending on where in the transcript you are. Over the past couple of years, there have been a number of methods that allowed us to look at alternative polyadenylation. So, for example, especially with the upcoming of RNA sequencing methods, which gave us basically access to sequence coverage across untranslated regions, a couple of researchers have developed computational methods that allowed us to interpret the differences in relative coverage for these alternative 3' UTRs. And just to mention a couple of those, there's the PARS, PECQR, or Kappa that allow us to analyze again a large number of existing RNA sequencing data sets and then make an inference about which 3' UTR isoforms have been used. In addition to that, there are also more tailored sequences or sequencing technologies that we can apply in bulk, where we specifically look at what is the exact end of a transcript and at the same time also try to quantify how abundant these are. So here again, there are a couple of methods, like, for example, a 3p-seq or a-seq, and all of them allow us, as I said, to look at single nucleotide resolution and also the quantitative information between those. However, these more tailored methods, they're usually a little bit harder to apply, and they're not used as widely as, for example, RNA-seq, as I mentioned in the first example. However, together, these have really shaped our understanding in how the choice of alternative cleavage and polyadylation has been made, and we learned a lot about which proteins are involved. And then more recently, researchers have also started to look at single-cell RNA sequencing. And again, here, we are focused on sequencing technologies that are inherently 3' end biased, like, for example, DROP-seq or 10x genomics or skyrNA-seq. And here we can interpret the relative coverage relative to known transcript ends. And then again, we can make an inference about isoform usage. In addition to these broadly used methods, there are also additional experimental methods like BAT-seq or single-cell polyA-seq, again, which may allow us to get single nucleotide information. But most prominently, researchers have focused on computational methods in order to infer naturally 3' biased data types and make an inference about which 3' UTR has been used. So we wanted to also add to this and study specifically how do certain regulators of alternative polyadenylation lead to differences in the choice of 3' UTR. And here we specifically turn to Perturb-seq, which is a method that allows us to combine pooled CRISPR screens with a 3' end biased single-cell RNA sequencing readout. So here we perturbed 42 known genes that we knew that were involved in the choice of alternative polyadenylation. And we targeted each of these genes with 3 to 4 different guide RNAs. And in total, we had a pool of about 178 guide RNAs. And we did this in 2 replicates and also in 2 different cell lines in order to get a robust understanding of our observations. So just to give you an example, so here we, for example, look at the gene locus of CBX3. And we identified 2 different peaks that are, and we can have access to the coverage relative to these peaks at the 3' end of this gene. And we see that if we group cells that have received a control non-targeting guide, and we group cells that have received guides that target NATI21, we clearly see shifts in the coverage from that indicate that the shorter isoform is used in the perturbed state. And we didn't only do this, as I said, for just one gene, but we looked at all our genes and we can recover some of the known regulators, like, for example, CPSF6 and NATI21 with both lead to shortening, and PCF11 and FIB1 L1, for example, with both have been previously identified to lead to lengthening of 3' UTRs. So if we cluster the counts that we identified in the peaks with the relative coverage, then we see that that our perturbations cluster relative to the complexes that have been described before. So, for example, we see that cells that have received perturbations for CSTF1 and CSTF3, which are both part of the CSTF complex, pretty much cluster together. And the same we see for the PAF complex and also for the CF1 complex, where we see NATI21 and CPSF6 together. However, we also noticed that while we look here at the counts in our peaks, we got very similar results when we just look at the gene expression changes. And therefore, we were very much interested in developing a new technology or a new computational approach that really helps us to look at alternative polyadenylation without the confounder of gene expression. And this is work that has been predominantly led by Maddie, so that's why I let her explain herself how she did this.
Okay. Thank you, Harm. And now I will be speaking about the computational analysis and method we developed in this paper. So as Harm mentioned, we use the 3' nature of this data to quantify a polyaA site counts matrix, where basically we're looking where reads aligned within different 3' isoforms, and we use this to generate a cell by polyaA site counts matrix analogous to what you would get in single-cell RNA-seq. However, as Harm mentioned, there's a lot of redundancy between gene expression and polyA site counts, and we needed a method to deconvolve these two. So to illustrate this point, I'm showing 2 examples looking at the 3' end of these genes. And in the example on the left, we have 2 polyA sites. And when you compare non-targeting cells to cells where PAF1 was perturbed, we see a large increase in the expression of this gene, but the relative usage between these 2 polyA sites is basically the same. In contrast, on the example in the right, the overall gene abundance between the perturbed and non-targeting condition is quite similar, but there's a striking change in the relative usage of these 2 polyA sites. For instance, this perturbation causes a large increase in the usage of this polyA site and a corresponding decrease in the usage of this polyA site. What we really want to focus on is relative polyA site usage. within polyA sites in the same gene. And if we had bulk single-cell RNA-seq data, we might just use a ratio, how many reads fall into this site versus this site. But because single-cell data is very sparse, this isn't feasible while maintaining single-cell resolution. So a number of groups have devised sophisticated computational strategies to address this problem in the past, and I just outline them briefly here. But most of them have relied on aggregated or pseudo-bulk profiles of cells that are in the same cell type, and then they use bulk testing methods like DEC-seq or bootstrapping to perform comparisons between cell types. Some papers have also imputed polyA site profiles from neighboring cells, which enables clustering. And I'll also mention that a lot of these methods pool information across polyA sites by calculating a gene level metric, for instance, proximal polyA site usage. But this gets rid of information about which polyA sites are specifically changing if there are, for instance, more than 2 polyA sites in a gene. So what we'd really like to do is develop a single cell metric that preserves both the single cell nature of our data as well as the site-specific information, which we believe is essential if we want to be able to look at local sequence content. So our goal for each cell and at each polyA site is to assess how consistent the counts we observe are with what we observe in a background distribution, which here we can estimate from cells that received a non-targeting guide. So for instance, from our non-targeting cells, there are 3 polyA sites in this gene, and we expect them to fall roughly as follows. We use the Dirichlet multinomial distribution to parameterize each gene using purely the non-targeting cells, and this allows us to obtain an expected value of the number of reads that will fall into each polyA site, conditioning on the total number of reads in that gene. For instance, if a cell had 10 reads at this gene, if this fell exactly according to the non-targeting distribution, we would expect the counts to we would expect counts like these. However, for instance, if we have a cell that was perturbed by NAD21, again, if there are 10 reads in that cell, we would expect the counts to be distributed quite differently. We need a way to quantify how significant this change is. So here's where we rely on the Dirichlet multinomial distribution, and that we can also estimate the variance of these counts. And from this expected value and variance, we can calculate what we call a polyA residual, which quantifies the relative usage of a polyA site within a cell. This has the nice property that if it's positive, it means a polyA site is used more than in our background distribution. And if it's negative, it means a polyA site is used less frequently than in our background distribution, adjusting for the overall level of gene expression. Going back to our prior example, again, in the example on the left, there's no change in polyA site usage. And this is kind of what we want to adjust for and remove as a confounder in our analysis. And in the example on the right, there's a large shift in polyA site usage at this gene. If we calculate the polyA residuals within this gene, we see as desired in the example on the left, there's no significant difference between non-targeting and perturbed cells versus in the example on the right, there's a strongly significant increase in usage of this polyA site. I'm just showing one of the polyA sites, but you also calculate a residual for all polyA sites within this gene, and you would see that there's one polyA site that has a significant decrease. Now that we have this polyA residual procedure, we can use this to identify significant changes in polyA site usage transcriptome-wide in each perturbation using a linear modeling approach. So we can identify genes where there are significant changes in polyA site usage, and we sought to characterize these genes, whether they cause tandem or intronic APA. As Harm mentioned, the majority of genes have multiple polyA sites, and these fall broadly into 2 classes. Tandem APA will affect the length of the 3' untranslated region only, versus intronic APA will also affect the coding sequence as you have premature cleavage upstream of the last exon. So we characterize the number of genes that cause both intronic and tandem APA for each perturbation. There's both variance in the number of genes that are affected, but also in what type of APA. We found that the majority of genes tended to cause primarily tandem APA in this darker blue color. But however, we also identified a subset of perturbations that tended to induce changes in primarily intronic APA. And we found that this set of genes was very interesting. Strikingly, all of these regulators of intronic polyadenylation that we identified have been previously shown to interact with RNA polymerase. And this is in line with previous observations that intronic polyadenylation is related to the rate of transcript elongation. However, what's interesting is that we did notice intronic polyadenylation in all of these regulators, but we also noticed that these occurred at distinct sites, suggesting that there are multiple regulatory mechanisms affecting the rate of intronic polyadenylation, and at which sites this occurs. And we're interested in exploring this relationship between transcript elongation and intronic polyadenylation in further studies. So looking primarily, shifting focus to just changes within the last exon or tandem changes, we found that most regulators tended to primarily cause either 3' UTR lengthening or 3' UTR shortening upon their perturbation. We also investigated the relationship between gene expression changes and 3' UTR length. Within NUD-D21, for instance, we found that the higher the degree of 3' UTR shortening, the larger the increase in RNA of the associated transcript. And this is in line with the idea that longer 3' UTRs contain more of these regulatory sequences that may impact RNA stability. So a bias towards shorter 3' UTRs will also result in a corresponding increase in gene abundance. And while most regulators had a strong bias towards either shortening or lengthening, we also identified a subset of regulators, in particular CSTF, that had a more intermediate phenotype, and it was more balanced between shortening and lengthening. To dig into this further, we considered genes that were responsive both to cleavage factor 1 perturbation, shown in these columns, as well as CPSF and CSTF perturbation. What was really interesting is that we found a subset of genes that we call module A, CPSF and cleavage factor 1 had opposite effects, but we also identified another set of genes that we call module B, where cleavage factor 1 and CSTF had the same effect. For instance, I'm showing two examples here. In module A genes, cleavage factor 1 perturbation induces 3' UTR shortening, but in CPSF, there's lengthening. In contrast, in module B genes, both of these perturbations induce shortening. And what's really interesting about this is that you have perturbation of the same regulator resulting in opposite effects in different genes. To understand how these genes behave in a little bit more detail, we also looked at the baseline usage of proximal versus distal sites in these genes, and we found that module A heavily favored proximal site usage, whereas module B more favored distal site usage. So, and this is without any perturbation. So, the fact that they differed in their polyadenylation site usage at baseline and in their response to different perturbations suggested to us that there must be something different about these two sets of genes, and we hypothesized that this difference was local sequence content. So to address and understand how local sequence content affects response to perturbation, we collaborated with Anshul Kundage and Johannes Linder. And in their previous work, they trained a neural network, Apparent2, that provides predictions of polyA site strength based on sequence content. So this identifies important sequence features that affect how strong a polyA site is, but importantly, we don't know anything about which regulators modulate this polyA site strength. They used both our perturbation data and Apparent2, their previously trained model, to train a new multitask neural net, which we call Apparent-Perturb. And this predicts polyA site usage from sequence in the context of each of our perturbations. This again allows us to identify sequence features that are important, but now we can link these sequence features to particular regulators. For instance, if we use motif discovery tools, we can identify important sequence features. In this example, we identified TGTA, which happens to be the canonical binding motif of NUD-D21, and this also occurs in the upstream region. Importantly, we identified this as important for NUD-D21 perturbation, but not CSTF perturbation, showing that we have some specificity of which regulator is actually mediating the perturbation response. Similarly, we also identified this downstream element, which is a known binding partner of CSTF, And we identified this as more prominent in our CSTF predictions. In addition to individual sequence features, we can also look at regions relative to the cleavage site. And we found that NUD-D21, this upstream region, was more important versus for CSTF perturbation. There's this strong peak at the distal sequence elements, but not at the upstream region doesn't appear to be as important. So in addition to exploring naturally occurring sequences, we can also use apparent perturb to engineer new sequences. For instance, if we add a second NUD-D21 motif to a particular sequence, does this increase perturbation response even further? And we found the answer to this to be yes, but this only happens if this second motif is added with a particular flanking content and if this occurs at a particular distance. We also identified a synergistic relationship between the canonical cleavage motif and this downstream element for RBBP6 perturbation, suggesting that there's some interaction with RBBP6 and other factors. So I'll advise you to read the preprint if you'd like to learn more detail, but the idea here is that we can link sequence features to specific regulators, and this allows us to respond, to identify both to perform both this linkage and also identify interactions between sequence features and perturbations. So our analysis can be expanded to any 3' single cell RNA-seq dataset. With this in mind, we also analyzed genome scale Perturb-seq dataset that was released from Jonathan Weissman's lab last year. And while they focused on changes in gene expression in their analysis, we wanted to reanalyze this data with respect to polyadenylation site usage. So we focused on RNA binding proteins only, and we found a couple hundred of RNA binding proteins that resulted upon perturbation in significant changes in polyadenylation site usage. We clustered these perturbations based on their polyadenylation site usage, and this allowed us to identify several modules of genes with similar behavior. Some of these modules involve members of the core CPA machinery, such as NUD-D21, as well as CPSF and CSTF complex members, but we also identified additional modules that may affect polyadenylation site usage directly or indirectly. For instance, we identified a module of genes composed primarily of genes in the exosome and PAX complex, which are part of RNA surveillance and degradation machinery. We also see genes that are related to ribosomal processing, suggesting that there's this relationship between the abundance of different 3' isoforms and regulatory processes throughout the RNA life cycle. With that in mind, I want to thank everyone in the CITIJA lab, as well as Harm Wessels and the Kundaje lab and Johannes Linder. And in conclusion, I hope that we've showed you that Perturb-seq can not only be used to investigate how gene expression changes in the context of different perturbations, but we can also use this to identify changes in transcript structure. We identified that perturbing these CPA regulatory proteins have varying effects on both intronic and tandem alternative polyadenylation, as well as 3' UTR shortening versus lengthening. And this was related to downstream changes in gene expression. We also identified that sequence is important in dictating this perturbation response, and we can learn this connection between sequence features and perturbations through deep learning. And also, we've shown that RNA binding proteins throughout the RNA life cycle will affect the abundance of 3' isoforms, and this occurs through both, probably through indirect and direct effects. So with that, Harm and myself would be very happy to take any questions, and really appreciate all of you tuning in and watching. And also thank you to CICA for having us.
You're welcome. Thank you, Maddie and Harm, for sharing your work with us. A really great talk. So we'll move on to our Q&A. If you'd like to ask a question, please raise your hand or type your question into the chat box. And one question that we have is, do you think that the ribosomal binding proteins that you tested directly interact with the transcripts to influence cleavage and polyadenylation?
Okay. We probably think it's more an indirect effect, that it's somehow mediating like the relative stability of these transcripts, but it's probably not influencing, production. Like there's been tons of work defining like which proteins are actually in the CPA machinery and which are kind of associated. So I think it's less likely that we have identified like a new CPA machinery member, but more likely that, something after that transcript is produced, there's differential regulatory effects on one isoform versus another.
Great, thank you. Well, if there are no further questions, I'd like to thank you again for your talk. It's been very exciting. And let you all know that the seminar has been recorded and it will be available soon on YouTube and on our website, which is multimodalintegration.org. You can also follow us on Twitter at segs_ICA or check our website for the latest announcements regarding upcoming talks and events. With that, I'd like to thank everyone for joining and hope to see you at our next seminar. Have a wonderful day.
Bye.
Bye-bye.